comes into contact with the stigma, thus completing the process of pollination. Conversely, flowers pollinated by butterflies are usually lightly scented and brightly colored, with nectar not very deeply hidden, while those pollinated by beetles and flies secrete a rotten odor. Flowers pollinated by birds, on the other hand, have large, odorless orange or red tubes which are rich in honey. Certain plants attract pollinating insects by offering them shelter. For instance, Amorphophallus, a 6-foot-tall flower, attracts pollinating insects by offering them a safe haven to lay their eggs. In the yucca plant, on the other hand, a female moth transfers the pollen of another flower to the stigma and simultaneously deposits her eggs in the locule of the flower's ovary. The larvae, which hatch out of the eggs, start consuming some of the developing seeds, while leaving enough of them to propagate the plant. Thus, both the moth and the plant are dependent on each other for the completion of their respective life cycles. However, certain birds and insects such as the bumblebee visit flowers for nectar and pollen, but don't bring about pollination. These creatures are referred to as pollen robbers. Honeybees, as well as other insects, also pollinate a large number of aquatic flowering plants, such as the freshwater lily. However, in certain freshwater plants such as Vallisneria and Hydrilla, and marine seagrasses such as Zostera, pollination occurs by water. This is also known as hydrophily. In Vallisneria, for example, the female plant produces a long-stalked female flower that reaches the surface of the water. The male plant, on the other hand, remains submerged in water, where it produces several tiny floral buds. Upon maturing, the buds rise to the water's surface and open up to expose the anthers. Pollen released from the anthers is carried by water currents, where some of them eventually come in contact with the stigma. In the case of Zostera, a dioecious seagrass, the male and female flowers remain submerged in water. The male flowers produce long and ribbon-like pollen grains which have a mucilaginous covering that protects them from getting wet. The pollen released in water is carried by water currents towards the submerged stigma, and thus pollination is completed. Surprisingly, in aquatic plants, wind pollination, also called anemophily, is more widespread than water pollination. Wind pollination also occurs in terrestrial plants such as grass, bamboo, coconut, and maize. These plants possess a compact inflorescence with well-exposed stamens that allow easy dispersal of pollen, and a large feathery pistil which makes it easy to trap pollen. The pollen, which is light and non-sticky, is produced in large quantities. A single flower of cannabis, for instance, produces 5 lakh pollen grains to compensate the loss of pollen associated with wind pollination. Thus, wind, water, as well as biotic pollinating agents bring about pollination. An important process that ultimately leads to fertilization and the production of seeds in plants. In flowering plants, pollination is an important mechanism that aids in transferring the pollen released from the anther to the stigma of the flower. If the pollen is of the right type, the stigma induces it to germinate and produce a pollen tube. The pollen tube, containing the 2 male gametes, then secretes hydrolytic enzymes which help the pollen tube to digest the tissues of the style as it travels down to enter the ovule through the micropyle, a minute opening in the ovule. Inside the ovule, the pollen tube encounters the embryo sac, the female gametophyte, containing 3 antipetals, 2 synergids, an egg cell, and a central cell with 2 polar nuclei. The pollen tube now enters one of the synergids through the filiform apparatus, during which it ruptures and releases the male gametes into the synergid's cytoplasm. Of the 2 male gametes, one enters the egg cell and fuses with its nucleus. This fusion, known as syngamy, or first fertilization, results in the formation of a diploid zygote. Meanwhile, the other male gamete moves towards the 2 polar nuclei and fertilizes them to produce a triploid primary endosperm nucleus, or PEN. Incidentally, this fusion is known as triple fusion, as it involves the fusion of 3 haploid nuclei. After the triple fusion, the central cell is called the primary endosperm cell. Did you know that syngamy and triple fusion together represent double fertilization? A phenomenon unique to flowering plants. Double fertilization is followed by several post-fertilization events which eventually transform the ovule into a seed and the ovary into a fruit. These events include degeneration of the antipetals, synergids, and the development of the zygote into an embryo and the primary endosperm cell into the endosperm by repeated mitotic divisions. Endospermic development is of 3 types: nuclear endospermic development, cellular endospermic development, and halobial endospermic development. In nuclear endospermic development, which is the most common type of endosperm development, the primary endosperm nucleus divides mitotically and forms free nuclei. This division is sometimes followed by cytoplasmic divisions from the periphery. In the case of cellular endospermic development, which is seen in plants such as utricularia, the division of the primary endosperm nucleus is immediately followed by cytoplasmic divisions. Halobial endospermic development, on the other hand, is an intermediate type of development where half the cells develop in cellular mode and the other half undergo free nuclear divisions. Interestingly, the number of free nuclei formed prior and post-cellularization differs greatly. Take the case of a tender coconut fruit, where the coconut water that you see is actually thousands of free nuclei. As the coconut matures, cellularization leads to a decrease in the quantity of water and the formation of the white kernel, the cellular endosperm. Therefore, in the coconut, we find both free nuclear endosperm made up of thousands of nuclei and cellular endosperm. Histological studies have shown that endosperm contains stored food in the form of starch or proteins, as in cereals, or oil, as in castor or coconut. This food provides nourishment to the zygote while it is developing into an embryo. It is interesting to note that the endosperm also forms a major source of nutrition for human beings. Incidentally, the development of the embryo, also known as embryogeny, occurs only after a certain amount of the endosperm has developed. In fact, in some plants, such as groundnut that belongs to the bean family, the mature seeds lack endosperm, as it gets completely absorbed by the developing embryo. When these seeds germinate, the cotyledons or seed leaves become the source of nutrition for the embryo. However, in mature seeds of castor, we see that the endosperm is retained as a storage tissue. In the case of cereals, the alurone layer, which forms a part of the endosperm, stores food as well as secretes amylase enzyme for the digestion of stored starch during germination of the seed. Studies by botanists have revealed that during early embryogeny, the zygote divides into two cells: the basal suspensor cell towards the micropylar end and a terminal embryo cell towards the chalazial end. The basal suspensor cell further undergoes mitotic divisions and forms 4 to 8 cells, which push the terminal embryo cell into the tissue of the endosperm to allow it to derive nourishment. Moreover, the terminal embryo cell also undergoes divisions to form a group of cells known as proembryo, which subsequently develops into a globular, heart-shaped and torpedo-shaped mature embryo. The structure of a mature dicotyledonous embryo is nonetheless different from that of a monocotyledonous embryo. A dicotyledonous embryo, for instance, consists of an embryonal axis and 2 cotyledons. The epicotyl, the portion of the embryonal axis above the level of the cotyledons, terminates with the plumule or stem tip. Below the level of the cotyledons is a cylindrical portion called the hypocotyl that terminates at the radicle or root tip, which is covered with the root cap. On the other hand, a monocotyledonous embryo, commonly found in the grass family, has only one cotyledon called a scutellum, situated towards the lateral side of the embryonal axis. The upper portion of this axis, known as the epicotyle, lies above the attachment of the scutellum. The epicotyle has a shoot apex and a few leaf primordia enclosed in the coleoptile, a hollow foliar structure. The lower end of the embryonal axis has the radicle and root cap covered in an undifferentiated sheath called the coleorhiza. Interestingly, the various parts of the embryo develop differently as the seed germinates. In this manner, the embryo, as well as endosperm, develops from the zygote and primary endosperm cell after fertilization has taken place in flowering plants. For us, fruit means a mango, papaya, apple, or a banana. However, for a botanist, fleshy and juicy mangoes or papayas are as much a fruit as are groundnuts or cashew nuts, which are dry in nature. Fruit, the final product of sexual reproduction, develops from an ovary post-fertilization. Incidentally, in most cases, fertilization and the development of fruit triggers the withering of other parts of the flower, including the sepals, petals, and stamens. However, in some cases like apple, floral parts such as the thalamus become a part of the fruit. Such fruit in which other parts of the flower besides the ovary are involved in the formation of the fruit are called false fruit. On the other hand, fruit like mango and papaya that develop only from the ovary are called true fruit. Interestingly, while mango and papaya develop after fertilization, fruit like banana develop without fertilization and are called parthenocarpic fruit. Moreover, in the case of banana, the edible parts develop from the inner tissues of the ovary, and the brown specks that we see are the remnants of the ovules. If you cut open a fruit, you will see that it is made up of two parts, the pericarp and one or more seeds. The pericarp is the fruit wall that develops from the walls of the ovary. The seed, the second part of the fruit, is actually a fertilized ovule. Typically, a seed is enclosed in a tough outer protective covering called the seed coat, which is actually the hardened integument of the ovule. The micropyle of the ovule persists in the seed as a small pore through which water and oxygen enter when the seed germinates. Inside the seed coat lie one or more cotyledons as well as the endosperm, embryo, and embryonal axis. The cotyledons, which are often fleshy and swollen, contain abundant food reserves. Endosperm, on the other hand, is a nutritive tissue nourishing the developing embryo. Interestingly, in certain plants such as bean and pea, the developing embryo completely consumes the endosperm. Such seeds that contain no endosperm are called non-albuminous seeds. However, in wheat and maize, a residual endosperm is seen as it is not completely consumed by the embryo during the developmental stage. Such seeds are called albuminous seeds. Sometimes, in addition to the endosperm, seeds also contain remnants of the nucellus called the perisperm that also stores food material. Such seeds are called perispermic seeds. Black pepper is a common example of a perispermic seed. Whether a perispermic or albuminous seed, as the embryo begins to develop, the seed starts to mature and lose its water content to become relatively dry. A mature seed, therefore, has only 15 to 20% of its mass as moisture content. Moreover, the embryo's growth rate slows, and under unfavorable conditions, it may enter a state of inactivity called dormancy, during which the embryo develops an increased resistance to desiccation. Conversely, conducive conditions that provide favorable temperature and appropriate water awaken the dormant embryo, and the seed germinates, with the embryo developing into a seedling. This ability of the seed to germinate is called seed viability, and it varies across seeds of different species. The seeds of spinach, for instance, remain viable only for a year or so, whereas muskmelon seeds remain viable for 5 years. Did you know that excavations around the Arctic tundra region have led to the finding of seeds of Lupinus arcticus, which remained viable for 10,000 years before germinating and flowering? Seeds, a character unique to angiosperms, also offer many advantages to these groups of plants. Seeds have special mechanisms such as bursting or wings that help them disperse to new habitats, thereby allowing plants to claim new areas to propagate. In addition, the seed's tough outer coat protects the embryo from hostile environmental conditions. Upon germination, seeds also have a better survival rate, as they are guaranteed nourishment by the cotyledons. As seeds are products of sexual reproduction, they help bring about genetic variations within plant species. Genetic variations are the key to evolution and also help plant species adapt and survive in different environments. Seeds, which help in the propagation of angiosperms, also form the crux of agriculture. Interestingly, farmers in recent times have started using hybrid seeds developed through artificial cross-pollination techniques. These seeds guarantee increased productivity and superior crop quality. However, when seeds born by hybrid crops are sown, the seedlings do not exhibit the hybrid characters due to the segregation of characters. This forces farmers to purchase new hybrid seeds year after year, which is an expensive affair. To address this problem, botanists are trying to understand the genetics of apomixis and transfer apomictic genes into hybrid plants. Apomixis is a form of asexual reproduction in which the seeds or embryos are formed without fertilization. In fact, seeds or embryos either develop from an unfertilized egg which did not undergo meiosis, or from the cells of the integument or nucellus. Botanists believe that converting hybrid crops into apomicts will prevent the segregation of characters in seeds produced by hybrid crops, and thereby eliminate the need to purchase hybrid seeds every year, as the seedlings produced by the apomicts will resemble their parents genetically. Did you know that plants such as grasses and those belonging to the family Asteraceae and Rutaceae are common examples of apomicts? Moreover, in certain apomicts such as Citrus jambiri, which belongs to the Rutaceae family, the nucellar cells surrounding the embryo sac divide and protrude into the embryo sac. Later, these cells develop into embryos, and hence a single seed contains more than one embryo. This phenomenon of having more than one embryo in a seed is called polyembryony.